welcome to this class. I'm Stephane Doyen, and online we have Michael Sughrue, and today we're going to present you our software. We come from a company called Omniscient, and we're presenting a software that allows you to do brain analytics, leveraging some of the latest work on machine learning. So a bit of content today. The agenda is we will first start with some of the design principles and history for the software. Then we will go into the neuroanatomical foundations of it, followed by an interactive demo. I hope you all have received your account and you'll be able to follow through. Then we'll move into some concrete applications of how we can use the software, and then we'll go over some of the future directions where we would like to take the project. So, in a nutshell, Omniscient is a cloud-based platform which is built for users from all computational skill sets and levels. This is a platform that is there to help you to work with some of the latest analytics you can find on the market for processing brain scans, whether this is for your research with others. We have built this in a way that you don't need to do any coding and you can access all those goodies. And just to give you a little bit of context, I years ago was a researcher in the academia in Europe. And I left the trade numerous years ago to move into professional software making. But about 2 years ago, I met with Dr. Michael Sughrue here on the line, who's a neurosurgeon, and he was explaining me all the challenges he was facing for some of his research and some of his work, and asked me whether we could find a solution to that. So I was pretty happy to go back to the field of neuroimagery and saw some of the latest advances in neuroimaging, the DTI coming on the rise, as well as advanced mapping of the brain. And I just thought to myself, this is absolutely fantastic. How can I get this to myself, and how can I bring this to Dr. Sughrue? I did a bit of research here and there, and what I found is a fantastic community, some amazing projects out there. Here's just a very, very, very short list of all the great softwares and projects you can find out there. But what I found as well is that the use of this software, it's still pretty daunting for someone who doesn't have a background in computer science or at least some elements of coding. And just to give you an illustration of this, this is a message I picked from one of the numerous ones you can find on some of the message board out there. And I've blurred the name of the actual software that shows this message because we don't want to speak specifically on that one, but just illustrate how things can be for the user. And as you can see, this specific user seems to be rather skilled, and explained that he's struggling to install the software because he has a message that something is failing due to a C++ compiler. Actually needs two, one G++ and one named Clang, which he both updated. So he had to work with the version of that, and he's running everything on Ubuntu 19.10. You can see as well that he's very, in a very helpful manner, pasting the logs here, showing, well, this is what's failing with my software. And I just thought to myself, that's actually a very, very powerful illustration of the state of matter of some of those software, which is, it requires a lot of skill. As you can see, the user needs install right on their machine. They need a C++ compiler. They actually need two of them. They also need to keep the versions up to date, run a special or an OS that's less popular than most of the users, and be able to use command lines. And this is something that is generally found in the in some of the academical software. There's some very good exceptions out there, but there is a reason for this. Generally, if you look at how software is made in the industry, it starts with extremely bright people putting together an algorithm which serves a function that we all need or want. And then what happens next is a lot of time is invested and effort through software engineering to actually turn that algorithm into a production grade algorithm, something that you can package, run, and repurpose in cloud computing environment. This algorithm gets deployed again by specialized resources onto a platform, and a UI is put on the top of it to make it all very helpful and usable for a user. So a lot of time is specifically invested into the development of this algorithm all the way to an enterprise grade platform. Additionally to this, an equal amount of time is spent on testing it, make sure it works in all sorts of boundary conditions and extreme conditions that users can apply to it. If you compare this to software making in the academia, what you find is that extremely bright people, extremely talented people come with a fantastic algorithm. And as you would expect, they publish about it. Even in better cases, that algorithm is also shared on a GitHub repository somewhere on the internet, which makes it accessible to everyone. That's all fine and great. But then this also brings some challenges. Essentially, what happens to that algorithm in terms of making it accessible to the user? What happened to the UI? Well, most of it is generally done through courses and tutorial, but really to bring it to a comprehensive software solution, that is something that is rarely seen in the academia. Only a few outstanding projects go there. So then, well, we thought about how can we work with that problem? And so looking at that user here again, there's three highlights I have here. You see that he has to potentially, if he wants to debug some of that software, he has to go into C++, that he needs some compilers and use some command lines. And we thought, well, let's do something here. Let's go back to what we should be doing, which is pick a language that is easy to access for everyone and talk to a wide array of contributors. And this is an important fact. That is, a good language is generally supported by a community, or the other way around. A good language is often promoted by a community. Obviously, every language has their pitfalls, but what we've gone for, at least at the core of what we're doing, is to look into Python. And so Python is a very interesting language. If you see back in about 2011, 2010, these are numbers from the mean active users you find on GitHub, is that at that time, Python wasn't so popular. What was popular, however, was generally MATLAB and R for statistical programming, which picked up very, very nicely in 2014. But somewhere around 2015, something happened, which is communities started to realize how versatile that language can be, in essence, that it speaks to statistical programmer, machine learning specialists, as well as software engineers. So there was a point of continuity. Aside from being very accessible, which is something that is illustrated by this cartoon where you see this character basically flying and another one asking him why he's flying, and that character says, well, it's easy in Python. All you have to do is to import the library fly. And that's where we derive the first principle of writing a software, which is Python at the core. And when you do this, what happens is that you connect an entire ecosystem of great libraries. So for instance, if you look at the ones on the left-hand side, you've got Nibable, which is great for using and loading medical imagery. You've got DiPy, which is absolutely fantastic for tractography and other type of analysis. But then using that common language, you're connecting that to equally great libraries such as Keras for high-level machine learning, or PyTorch for other machine learning project, NumPy and others. The wealth of this ecosystem is absolutely staggering. So Python at the core. Second thing is, well, let's look at another set of issues that user is facing. He needs to control for the version of the compiler he's using, or at least he has updated them, which is probably not something explicit, but he tried as a point of diligence, as well as running Ubuntu 19.10. And this is all fine and great and can be very useful. But on the other hand, what you're asking to your user when you're making such software is for them to control the environment in which you've developed the software so that it runs in the same way. Well, obviously, this can create a problem because every user has different machines, different access, different environments. And that's where we derive our second principle, which is containerize, ship, and repeat. So if you take a Python core and a project we're using or a repo structure that is called a cookie cutter, it's publicly available, you can basically start to put things together in a way that becomes quite standardized. So it kind of acts like an SDK or a self-development kit where you can start to put your algorithm together, and then using Docker, which is a system that packages your application with the environment it needs to run. You can standardize your algorithm for deployment on the platform, and you ensure that it runs exactly the way it should. And that's our second principle in building our software here at Omniscient. The next part is, well, let's carry on with that example. So if you see this user needs install write on his own machine. And this is also an interesting part because as you know, processing brain images is extremely demanding for your CPU as well as other computer resources. And this shows that there is a solution to this, which is the use of cloud computing resources. And here's a question, right? Why would you use your own CPU when you could use thousands of somebody else's in the cloud? Common issues, for instance, you find with laptops. So if you look here, let's see, you've got an 8 hours processing time to get the perfect map over that latest scan you acquired. Well, what happens if your computer has pushed some updates? Then you find yourself that you have to restart midway, and then it just breaks the process. You have to start all over again. Another common issues you find with laptop is, let's say, well, you've got a multi-core laptop here. What happens if the software is not optimized for it and you just run it on one core? You basically have 3 cores idling. Or the other way around, if you use your 4 cores, then what you do in terms of being able to work on your machine, you almost have to dedicate your laptop. So cloud computing, to a certain extent, solves this by allowing you to, at a few clicks, spin instances that I have here in this example about 48 cores and gigabytes of RAM. And that's where we derive our third principle, which is, well, how do we get this? Microservice everything. And so the way this works is with a combination of technologies such as Docker with the programming language Python, you can use something which is called Kubernetes. And Kubernetes is a great technology. It takes your Docker container and allow you to spin instances or pods in the cloud on demand and start to do heavy lifting in terms of training models, crunching numbers, looking at different metrics. And that is extremely powerful. It creates software that are highly versatile and very, very convenient and resilient in the cloud. So I'm going to go back to my initial question, that my initial quest, finding a way to get this DTI tractography imagery and this amazing brain mapping available at the least amount of coding or technical specialty to Michael Sughrue and following the public. Well, my answer to this was, well, basically we have to shift gear here, and we have to build this cloud platform using modern software architecture. So these are just 3 principles we apply here, but obviously there are a lot more, and you'll see that throughout the demo. And some of which here that I'm listing, which we found essential for us here at Omniscient. We went over the 3 first ones. Another one, obviously, which is extremely important, and you'll see that in the next stage of the presentation, that is the end users are part of the development cycle. So we sit with our end users on a regular basis, more than twice a week, to make sure that whatever we're crafting here is actually properly serving their need. Additionally to that, the software obviously needs to be anatomically specific, and that's something we'll cover very shortly. Fast, the software needs to be fast, and that's quite important when you think about processing data at scale. It shouldn't take you several hours to get a single scan. You should be able to process many scans in the shortest amounts of time, and we'll see that. Other principle we need to have in the software and that we've integrated is the ability to work with clinical grade imaging, which, as you may know, is not often the quality you find in research grade imaging. Big data, meaning that we want the users to have access to the largest library of data they would want, so that they can run all sorts of research that they've ever dreamed of. And then eventually, we also use machine learning at the heart of our system to handle the difficult steps. So machine learning is used wisely across the platform, not everywhere, not as a standalone, but really where it requires specific steps that are difficult for the user to handle. And these are the principles we're using. So now moving into a little bit more of the neuroanatomical grounding, I'm going to hand over to my colleague, Michael Sughrue.
So thanks, Stephane. I think it's a good introduction. I'm going to talk about, and as Stephane pointed out, I've been part of the development team as much to the abilities that I have as a neurosurgeon since the beginning. And what we aim to have is to solve a problem that immediately struck me when I saw some of the great work that people were doing, which is that we needed to put neuroanatomy back into neuroimaging. Now, what do I mean by that? So if you go to the next slide, if you look at diagrams like this, and again, you can find something like this in a paper in about 5 minutes if you look for this, or even less. It's not hard to find examples of this. If you look at the left diagram where somebody's showing a map of how different brain areas are talking to each other in some kind of function, or on the right where you have labels, and in this case, these were labeled as the parietal cortex and the temporal cortex. What both of these have in common is a relative lack of anatomic specificity. And again, for example, if you look at those circles, probably this is not exactly how the brain was activated, particularly that some of these straddle to both sides of a sulcus. And furthermore, it relies from the idea that people are thinking about this in terms of cognition without thinking about the fact that there is a part of the brain that is probably fairly sizable that is doing this. And if we're not anatomically specific about what we're saying, we're giving up a lot of ground that we really should be leveraging. It makes it difficult for us to communicate with each other about what we found. It makes it very difficult to repeat things and to be certain that we're finding things in a consistent way. And most importantly, neuroanatomy is the foundation of clinical neuroimaging. Certainly in my field, it's absolutely essential, but it's also in neurology, and eventually, we hope one day, psychiatry, where what part of the brain you're talking about is extremely important. If it's not grounded in anatomy, then we can't determine how lesions affect that anatomy. And if we can't have a way of communicating across studies where we're all talking about the same thing, we can't iterate, we can't improve, we can't test hypotheses. So we've built this into the software through work that I've done for many years, leveraging what a lot of other people have done. Again, this scheme is heavily grounded in the Human Connectome Project scheme published in Nature in 2016. It's not to say that this is the only possible atlas we could have, but it is a pretty good atlas if you've read the methodology. And it utilizes a lot of things we're familiar with, so it's learnable. It has Brodmann areas in it. It has visual V1 cortex. So, and for example, most people know what area 4 means. And so you're not starting from scratch when people become familiar with this, some of the naming in this. And so even though there are a lot of new areas, it's not completely foreign. And so when we work with other atlases, we attempt to try to translate as much of the data as we can onto this atlas, so that if we're talking about area PF, for example, we're all talking about the same basic place. Next slide. One of the things when I saw this paper, I immediately started working on how do we get this to work in the real world. And in order to make this work in clinical medicine, we have to understand this in terms of anatomy. We have to be able to have ways for physicians and people who are clinical researchers to translate the neuroscientific findings of or basic researchers into something that can be acted upon. So we published about 18 papers. We put this in the neurosurgical literature. We did that on purpose. We wanted to make sure that neurosurgeons knew that connectomics existed, for one thing. Most of them had never heard of it. And furthermore, that we could take the atlas that we were dealing with and explain the connectome in terms of that atlas, which is a fundamental basic building block of using this in the real world. We go to the next slide. So what we did in these papers was we basically first focused on going back to cadavers, finding where these areas were in the brain, and to demonstrate them in visual ways. We also went and used tractography and resting state methodologies inside of the HCP software, as well as other software, to make a map of every individual area, all 180. And one of the things that strikes you when you really start to get into where these areas are is many of them are in some very tucked in clefts of the brain. For example, if you look on the right, there's a number of areas on the undersurface of opercular cortices that are different than the outer surface. And this really highlights that the fact that brain anatomy does not care what we call a gyrus. It puts the function where it thinks it needs to put it. And it views these little nicks and crannies as equally valid places to organize function as areas that are easy to understand. And so it's incumbent upon us to be very specific of what we talk about, or it's going to be very challenging for us to translate anything that we find into something that will help a patient one day. We then began to use the task-based literature to annotate this connectome. And a lot of the data that we utilize is built into the software. So we utilize coordinate-based meta-analysis, which is a well-established research technique to make activation likelihood estimates, as you see on the left, to tie this to the likely parcellations of interest, and then using diffusion tractography to build estimated network maps of what we thought, for example, the language network here looked like. And there's a wealth of knowledge that can be gained by combining these techniques and painstakingly looking at the areas to determine across studies how do we combine prior information to link it into usable constructs. Skip this slide. Also, it's important that when very good work is done, for example, this is an atlas that many of you are familiar with from Yeo and Buckner in 2011, where they were able to utilize resting state to divide the cortex into various combinations of networks. Here's the 7 network parcellation that they did, and this has a lot of value. But one of the things that we focused on was how do we map this onto a scheme that we know a lot about, because we've done a lot of mapping to this. And recently we've been able to translate a lot of the findings from this atlas into the same architecture of Glasser, the Glasser atlas. And we leverage that in our software as well. And for those of you who are not familiar with this atlas, there are the 3 very familiar visual, sensory, motor, and limbic networks, as well as the more expansive DMNC and extended dorsal attention networks that you see here. And again, this is a very useful way to think about the brain. It provides a different view on the brain. but it's important that we keep the nomenclature relatively consistent so that everyone become familiar with a single naming nomenclature. Next. So, Stephane, okay. So what I'm going to do now is I'm going to take over the screen, and I'm going to quickly walk through the viewing platform on the software. And if you have a password and are able to log on, it's would be helpful if you followed along. I'll try to go slowly. So what you can see is that our software is 100% based in Chrome. There's no installation, and it views in a web browser. So basically, it involves logging onto a website. And what you can see here is this is the 3D rendering. There's also 2D renderings here that we're working on getting a compelling visual translation. It's very important to us that the visual renderings are understandable and useful and user-friendly. So we tend to test things very extensively before adding them. As you can see on the left and right side are navigation panes. The right side involves mainly controlling what's in the rendering and how it's viewed. And the left side involves subsetting. And we'll talk a lot about how to subset different combinations. Also, as you can see, there's a variety of different tools here on the top. These two will be coming relatively shortly. This is a mainly DTI-based module that works with a machine learning-based atlas that we'll describe in more detail later, but I'll demonstrate its basic character in this demo. This is a series of resting state correlations. And then also in a connectivity matrix, I'll be demonstrating that. And here at the guide, this links to a series of a lot of the anatomical and literature-based descriptions on what these areas do, or at least what we know about what they do. So to quickly run you through the right side panel, because this is probably the first one to understand. And for those of you who are not extremely familiar with working with three-dimensional renderings, these are the traditional three-plane orthogonal 2D image views that are all linked together. So for example, as I work with sagittal, you see that things start to disappear into the page as they reach the plane that they're located in. It's important to note that that's going to always cause a challenge with parallax, so just be aware of that if you haven't worked with this before. And you can see we have different views that allow us to really more precisely determine the position in space. Running to the next one, this involves a very simple tool for selecting tracks, turning them off and on, for example. This one here determines whether or not the object on the screen is showing tracks just between the parcellations and gray matter objects that are selected, or whether or not you want to show all tracks going into and out of an area. There are reasons and situations to do both, and I'll show some examples. The parcellations involves an atlas that's displayed in three-dimensional space, and what your options include a fitted atlas, which is a generic HCP atlas that's been affine warped onto the patient's brain, whereas the conservative and liberal are machine learning based atlases, meaning that we have utilized a technique that we'll describe in a bit to personalize the atlas using the structural connectivity to that patient. Scrolling down, the connectomic, I'll show you this in a little bit, but this is for working with the resting state correlation data. ROI selection is for making customized tools. And the targets for export will not be available on the research grade software, as this is really a clinical application, and it's designed to export resting state, targets for either transcranial magnetic stimulation, surgical planning, etc. Down here at the bottom, if you see download case, this is the link to all the derivative files. And Stephane will cover this in more detail, but it has a collection of different nifties, all of the atlases that are created, the tractography data that's created, and as well as numerous adjacency matrices for functional and structural connectivity that are automated for all of the atlases provided, as well as structural graph theory data and some of the anomaly detection tools. So it really is, with about an hour pipeline, a wealth of data that's automatically created with every upload. And we'll go into some detail, but again, it basically essentially is a very high yield version of what you would get in processing something through FSL. On the left-sided panel, what you see is, and again, this panel is constant regardless of what mode you're in. So because it allows you to either segment by network. These are based on the meta-analysis work that we've done of trying to tie parcellations into brain networks. Here, you could actually call individual parcellations. We'll go through this panel. Here we have white matter bundles, and what this is showing you is all the areas that are connected through a specific white matter bundle, such as the frontal ascending tract, the superior longitudinal fasciculate, the uncinate, etc. And you can even look at particular regions, so it's possible to call the entire DLPFC if one wishes, and we'll kind of demonstrate how to work with that. Now notice that this will also be the same tool that we use in the connectomic model, or connectomic module here, and notice that you'll see that these two systems are completely linked. Now, let's start with something simple. So when I hit the language button, what I tell the software to do is to bring back the parcellations that are typically associated with language functions based on the meta-analysis and the tracks that go between them. So if you see there, and it is possible for some of the clinical applications to export this to a single object. For research, this will not be activated, in part because that's really a clinical application and has to go through regulatory boards in general. Now, if we were to look at this, again, we can apply different atlases to choose which parcellations get picked. Or it's even possible to say, I want everything that goes into and out of the language parcellations. Now, this is going to get pretty noisy, but there you go. You can see how much of the brain talks to this. Now, notice that the resolution of crossing fibers that we're able to show is quite substantial. And for those of you who have spent a lot of time with diffusion tractography, and mainly DTI, you'll notice that CSD is able to really show quite a few more fibers. Now, again, it's generally recommended that if you're making objects that you clear the screen between this. This brings you back to the default. Now, again, going into this atlas, you'll notice that this atlas does not have smooth boundaries. And the reason for this is that it's based on the patient's tractography data. So to give you an example of how this works, we start with a non-machine learning atlas as our prior. We use this to restrain some of the connectivity, the machine learning, to prevent it from looking in crazy places for the Broca's area. So we don't expect the Broca's area to be in the occipital lobe. And as you see, it starts with an atlas like this that's relatively smooth. It's got clear, nice boundaries, but it's probably not reality. Some of those voxels, if you've ever spent any time with atlasing, will inevitably be in sulci, the Sylvian fissure. And one of the challenges that we found is when we were trying to apply this to patients, it would put it into tumor parcellations in clear areas of necrosis or resection cavities, or things that couldn't possibly be true. So while this atlas is fairly standardized and non-controversial, it's not really reality, and it's not personalized. So when we basically run through the processing scheme, what it will do is it will look at the tracks and try to determine what's the best fit for a parcellation of every voxel in the mapping. So it will look at, for example, around here and say, are you a voxel of 44? And it will assign it yes or no based on who you talk to, meaning your structural connectivity. When you do that, you get a little bit more noise, obviously, because it's based on data. But what we found is that it reclusters about somewhere between 3 to 5% of voxels, depending on the individual. And we felt that that was pretty good. We haven't seen very strange results with this. For example, we've never seen it put the motor cortex up here. We've never seen a situation where a normal person was missing parcellations. But note that this methodology, and again, this is a normal individual, has the ability to tell you if it can't fit parcellations. One of the things we're working on now is if a parcellation is there but is significantly diminished in volume compared to normal, but that's a different machine learning tool necessary to stack that in there. But what it will tell you with fairly good confidence, and I've tested this on hundreds of patients, is whether parcellation is completely absent or not. Now notice that we have a much bigger variety of areas, and so to run through a few of these, for example, we have the default mode network, and as you can see that the machine learning is fairly good at making a convincing location for the DMN and showing its basic structural connectivity. And if you get into areas such as the medial temporal, we have modules, for example, and if you look at the of the entorhinal cortex as well as the amygdala and hippocampus. In the subcortical area, we map the big structures. We're working on making a more detailed subcortical atlas. Again, we were very impressed how well it did with finding basal ganglia in general. So we didn't go for the win, but we think that given our understanding of this atlas, that we can get probably very compelling subcortical resolution, just because we have more experience with this now. And again, we have different combinations. Now, again, looking at how to leverage this. So if you look at the sensory motor map that we make, it's again going to cover not just the motor cortex, but it's going to cover the supplementary motor area and all of its subparcellations. It's going to cover the cingulate motor area. Now, you might decide that you only want to see the SMA. Well, that's possible to do. You might decide that you only want the right. It's possible to do that too. So, for example, if I were to choose this, what I've done is I've told it to turn off all the other areas and only show me the SMA. And as you can see, once I do that, the frontal acyclic tract disappears. But if let's say I wanted to just see how the SMA contributes to the corticospinal tract. Well, right now, I have this in the situation where the tract, the between parcellations, is on. I could turn that off, but it's going to show me some things that aren't corticospinal tract. So how would I handle that? Well, the way you can do this is you go to an individual parcellation and you click brainstem. And if you click the brainstem, what you did is you added the ROI that is the brainstem. And now what it's doing is it's telling everything that goes between the SMA and the brainstem. Now, if I wanted a pretty picture of this, I could also just turn the parcellations off, and there you go. There's your tracks. And let's say I, for example, didn't want all these small U fibers. I could increase that, and fairly quickly I have, a nice bundle if that's all I wanted. So as you can see, that the system is extremely flexible in what you can generate for tractography, but more important, exploring tractography and exploring it in a way that's scalable to patients with disease. Now, let's look at another methodology for subsetting. So here I'll just call the DLPFC.
Sorry, Michael, just one very quick comment here. Thinking, you mentioned pretty picture. And just for those that are viewing the course through the video, essentially I'm also viewing it through the call-in system here. And generally these things have a refresh rate. So what you're seeing on the screen seems a little bit laggy and jittery, but it is because it's actually run through the video conference system. So hopefully you have it running on your laptop. And I can tell you that on the most common laptop, let's say i5 processor with about 8 to 16 gig of RAM, this is 30 to 50 frames per second. So it's extremely smooth and quick moving and navigating the different parcellations and tracts.
So as you can see here, we have the DLPFC. We can turn parts of it off if you'd like. And this will come into handy when I show a trick that many people who are interested in TMS have brought up as an interesting strategy. So as you see, I turned off one side. I can use the coronal to actually get this into the brain, as you should see here. So it's not floating out in space. And as you can see, we've set this up where you can pretty much do lots of things with this. Now moving over to the connectomic panel. So let me just show a very simple method of how this is used. And so as we come onto the screen, let's say I want to see how the central executive network talks to the default mode network. This is a very common resting state question. What I did is I just generated that analysis. So as you can see here, what I have is a list of the parcellations that we've mapped to the CEN versus a list of parcellations we mapped to the default mode network. And unlike most connectivity matrices, you can actually interact with this one and look at specific numbers. So for example, this is showing you the left area PSL, which is the Parasylvian language area, and left area 46 have this correlation value in our anomaly detection. Now, I'm going to go into a little bit of what this is actually showing you. So if you go on to the connectomic first, you can see that you can actually change between raw and anomaly. So this is sort of the raw data, and so this is a series of just standard full correlations. And as you can see, these little dots are telling you that this is part of the CEN. And what we found though is that, again, looking at this, everyone has seen these pictures and papers in the corner of different things, but this is not very usable. It's not very it's not really simple for humans to look at. So just generating a correlation map and putting it in the corner of a paper is probably not going to be what changes medicine. What will probably change it is our ability to look at that and find what is abnormal and to know how to subset the data in ways that are meaningful. So as you can see here, what this is a tool that we've created, and this is generated in the folder as well, where we look at, we created a training set of about, and Stephane can correct me, but I believe it's about 300 normal individuals for different data sets on different scanners. And we determined what the standard range of correlations for these normal, or at least people without disease, was for all the correlations we perform, which is I think about 143,000 tests. And what this will give is an output of one of four things. So either it will show you a NAN for areas that are high variance in normal individuals, and thus we felt that we shouldn't show it because we didn't want people to fixate on what might be statistical noise, given that we have a massive multiple comparisons problem every time we do connectomic data like this. White would be within normal limits in an area that is not high variance. Red are areas that are above the, in this case, the 3 sigma threshold that I've set, and blue is less than. And so one of the ways that we've thought to use this for example TMS targeting is, for example, if you notice that there are some of these columns that have lots of anomalies in this. So whether this is the best target is a clinical and research question. But one of the things that is very useful is we can pull that target in 3-dimensional space as an ROI. So see, that's what we just called up right there. And so it's very seamless for interworking with, well, what's abnormal? Now, another strategy for targeting TMS, and again, not saying that these are the right strategies, these are just strategies that have been described and have data behind them and make sense, at least to my head, is looking at the DLPFC and studying its anti-correlation with area 25. So what you can see here is if I go here to 25, a second here. Promise is in here somewhere. Well, in the interest of time, let's see that subcallosal 32 is 25. You see that it's very simple to basically look at how this is anti-correlated with the DLPFC. And some people have recommended that looking at the subcallosal cingulate, specifically area 25, is one methodology for targeting this. So it's possible. Oh, there we go. There's area 25 right there. So again, this is an infinitely flexible platform. Now, let's say, for example, I didn't know what these things were because I'm new to, the scheme, or I don't, I haven't sat there and memorized this like I have. If you click the guide is also synchronized to this, and it's possible to say, okay, let's say I want to know what is this FEF. Well, there you go. There's an entire summary of the tractography, what the resting state correlation in the HCP data suggests, and what the white matter and correlations, and more importantly, the boundaries, who it's talking to, and what we think it's doing. And again, this is synchronized. So if I add V1, all of a sudden I'm going to change what's in V1 will show up in this thing, in this menu. Looking at one more thing before we move on, it's possible to look at how subcortical areas talk to or correlate with cortical areas. So notice that we've mapped subcortical areas into the scheme. So it's possible to look at, for example, basal ganglia structures, the diencephalon, etc., and look how it's for example, talking to the salience network. So again, it really is simplified working with data. Now, again, Stephane will talk about this folder, which again is a cache of data of different types, but how do you put files in here? So there's a variety of different ways to do this, and we'll kind of go through this. The load local file allows you to take one of these download folders and view it without having to go through the upload processing. But again, it has to be in the format that we export in. So you can't just put a nifty file in there and load that. It won't work very well. This is an algorithm that has a specific way that it reads the folder and what it expects what the names to be. But if you want to upload a new case, we have the ability to work with DICOMs, with nifties, and even to actually API integrate into packs. These are obviously for different users in different situations. One of the things that this does, again, is if you were to point this to a folder of DICOMs, it performs the DICOM to nifty step for you and de-identifies the data. It's actually possible with an API integration to use your own packs to re-identify it if you were doing this for treating a patient. But given that most people taking this course are probably researchers, it actually does all your anonymization for you as well. It is actually, we have multiple safeguards that prevent us from transmitting PHI of any form. And so this can really simplify your workflow by not having to do your own nifty conversions. But if you're working with nifty data, this has the ability to work with this. The only thing is that you have to name your own case. So as you can see, we try to make this as flexible as possible to get data in and out. And then I'm going to hand this back to Stephane and let him take back over.
Thank you, Michael. Let's see this thing. Okay. So Michael mentioned the ability to work with Nifty. If you want to upload Nifty to the platform, we recommend using that DICOM to Nifty converter. Make sure you use the latest version. Previous versions create different types of Nifty that aren't so compatible. Okay, so moving on. Just the next thing is just to give you a peek on what is under the hood. So to produce the results that Michael has shown, there's a series of things that are going on. Essentially, when you upload a scan, it goes through this microservice infrastructure and get processed by a series of microservices, Python-based microservices that are doing a series of transformations and computing operations. And so amongst which we find that we have basically, we generate tracts using CSD, so constrained spherical deconvolution. We also use gradient distortion. So that's a feature we have, and that gradient distortion is applied both on the bold signal, but is also applied on the DTI signal. We are using motion correction as well for correcting with acquisition error or subject moving during the acquisition phase. We are obviously cleaning our bold signal with ComCore as well as other in-house algorithm. Another important type of correction we're applying is basically a DMA correction. So if you have patient scans that have free water in them, this gets canceled out from the DTI imaging. So that's basically the short list of everything that's happening under the hood. There's obviously another set of proprietary algorithms running in there, which produce the results you've seen. Okay, so moving on to specific applications. So Michael will cover some more clinical applications there, but before we got there, I wanted to take you through some of the potential research applications you could get to using the software. So we really thought about the software and how we made it to empower researchers with great tools. And Michael walked you through the viewer, which is extremely nice for looking at patients' postulations or subjects' postulation scheme and different tractography elements. But a lot of the research actually starts at that step, meaning this is the point in time where you have data in a usable format or in format that you can start to interface with what you're trying to get to. And this is why you must have seen in the UI, we have this button here, which we call download case, or that download your subject right there. When you do this, what happens is that it will go on the server and create an archive for you. That archive is for standard case about 600 megs, and has all the files that have been generated by the core assets of the platform. This is available for you to download so that you can start to interface with your actual scripts and analysis. And so what you find in there is basically a series of things. So you have the list here on the right. The important part is the input ones. So you're getting your inputs converted. If you were to work from DICOM, you're getting them back in Nifty compressed. And these are all also co-registered. So they all work together in the same affine system. What you get as well is a series of anatomical elements. So you get your T1s, and then you get the atlas. So the raw atlas that Michael has showed is the fitted one, and the one that has been corrected by machine learning. They all come back to you. You don't see an extension on this tree view because this is actually a folder. In that folder, there's a series of further files that you can load and use in your IDE, whatever language you're using for your analysis. You get the tracked files as well when you want to run analytics on some of the tractography imagery and bulk process hundreds and hundreds of scans. That's for the anatomical and the structural. You're also getting functional data. So you're getting the bold process signal, as well as the correlation matrix computed for you, which you can in the shape of a CSV. Based on the machine learning atlas or the extrapolated machine learning atlas, or even the generally fitted machine learning atlas, sorry, non-machine learning atlas that Michael demoed. Additionally to that, we have this feature as a preview in the product. It's not available yet, but the data for it is available. Is that any scans you're passing through the system is turned into a graph. And the graph here is showing basically an adjacency matrix with postulation to postulation and tracks count between the postulation as an edge count. That's about it in a nutshell. And so what I will try to show you here is how you can use this thing. So I'm just gonna switch out of my presentation. Hopefully you guys can see my screen there. And so what I have there is basically this folder that you can download for each and every single one of the scans you're uploading. And in there, exactly what, as you can see on the right-hand side, I've got all my goodies. It's really a wealth of data. And if you want to conduct research with this, you can upload all of your scan, get them processed. They come back, and you can start processing further your scan at massive scale. And here's a short example where I've created this case. I've downloaded it back from the platform, and I thought, well, I'm gonna run this here. This simple script, it's just a few lines in Python, and as you can see, I'm not calling many libraries. But what this does in two simple functions based on the data that I've downloaded, it computes the volume of each and every single postulation in cubic millimeters. Okay, so that's all the code it takes based on the data that's been processed. So all the heavy lifting and the complicated pre-processing has been done for you. It's there and available in that folder. All you need to do then next is just to create these few lines of codes to start reprocessing it to add to your research paper. And I'll show you here. I've got this this folder here, and I'm just going to run this code there. So re-triggering this line, this will call my code, run my volume analysis function here, and just getting back to this folder. What I have now in just a few clicks and a few lines of code is I now have a postulation ID. We have a lookup table to give you what the name of these postulation are, the number of voxels that are part of that postulation, and then the volume of each and every single one postulation. So if you're running a research, for instance, where you testing and retesting subject, you want to compare between two treatments, if there is any kind of change in postulation, well, you can do that, right? You can basically run the subject once, pass it through the platform, get it back to you, run that analysis, and then test the subject again, run the same analysis, compare it. The same applies with basically everything else that is available here in this bag of data. Let's say you want to see how the adjacency matrix could change from one test to another. The same thing apply. You test one subject, you acquire the data, you push it through the platform, download it back, run your analysis, and off you go. So really a lot of powerful tools available for researchers, and a well worth of data for your research paper. Okay, so I'm going to pass it on now to Michael here, who's going to walk us through some of the more clinical applications that can be thought of when looking at this information you're getting from the software. Over to you, Mike.
So again, we aim to make the software able to do a lot of things, but one of the things that we wanted to bring back into connectivity-based research is the ability for clinicians to be part of the equation without having coding experience or professional programmers on their staff, et cetera. And so there's a lot of potential applications. One of the ones that, and how I first got interested in the field is, I'm a glioma surgeon, so I take out brain tumors for a living and do a lot of awake surgery. And so as you can see, when we first got interested in this many years ago, I wrote this book, and as you can see from the cartoons, these are different ways to remove gliomas in different parts of the brain, preserving specific white matter bundles to minimize cognitive damage. One of the challenges is that these are the obvious ones. So for example,
Sorry, Michael, I think your audio is gone. Yeah, we missed the 3 last sentences. Your audio just went a little bit wild there.
Can you hear me?
Yeah, it's better now. I think you can go back to about 3 sentences.
So, but one of the things is that these are aimed at things that we know, or we're fairly certain we know, are bad ideas. For example, cutting the arcuate fasciculus. And one of the things we want to do is empower people to fill in the void of what do we not know that we don't know, of what is a bad thing to do in surgery. But obviously, as we gather data for surgical applications, it can inform our surgeries and have immediate impacts on improving patient outcomes. So if you look at this situation, so next slide, Stephane, that where you're working in the frontal lobe, you can utilize connectivity to tailor your operation to avoid certain bundles. And because again, in glioma surgery, we don't have landmarks. We're basically cutting an amorphous object that has incredibly important microstructure. Next one. So one of the things that we've worked with the software is providing maps of not just tractography, which can be very difficult to discern what is important and what is not important, because we have to cut the brain to do an operation like this. But more importantly, to annotate the connectome and provide it in ways that are linked to functional networks. So we don't know in this large frontal glioma if what the default mode network that's being mapped there, or the central executive network, are behaving like they would in a normal individual. That's a scientific and clinical question. What we do know is that it's giving you fairly plausible and believable data on where the likely locations of these networks are. And how we would utilize this in surgery is to make cuts like we're demonstrating here, and then if you advance it, and here, to safely remove this tumor. If you look at the parietal cortex, next slide. Again, this has different goals and different mandates of what you're trying to avoid, particularly if you're dealing with a large medial parietal tumor. And so, for example, the montage in this case would be different. Next slide. And you know, for example, this large parietal butterfly glioma is, if you were working on removing this, has risk that we weren't aware before connectivity that was there. So for example, next slide. Even though we would reasonably be certain that we were near the motor cortex, and probably know if you were sophisticated that the language system was at risk laterally, what we wouldn't know, as what was on the next slide, is that this tumors wrap around the default mode network. Again, we don't know that in this patient that the DMN is working like it normally does. In fact, we know that there's dysfunction, and for a long time we didn't understand how losing your splenium to a tumor would cause profound memory and behavioral problems. Now we have a better understanding because of the improved understanding of the anatomy of these tumors. But again, ultimately, glioma surgery and understanding glioma surgery comes down to making safe cuts. So if you just advance real quick a couple times. So even though these are a complicated set of cuts in three-dimensional space, again, this is what we're trying to accomplish with doing a safe surgery in this situation. Next slide. Another potential application. Oh, sorry, go back. Actually, that was a good slide. Sorry, go for it. One of the things we're working on and preparing for FDA submission is a clinical application of this, and what it's aimed to do is to eliminate the need for human input to generate believable maps of networks that are surgically specific. So in this case, if you were to select the quick plan of surgery module, the next one you would get to is a list of operations, so that this reduces the complexity of making believable brain maps to selecting, for example, that I want to be in the frontal lobe, and it would then go use machine learning to generate the export of different networks that are relevant to this, that could be exported to DICOM files and put on surgical navigation. For example, this is the language system. Another clinical application, which I'm sure many people who are familiar with here, is transcranial magnetic stimulation. It's an FDA-approved treatment, approved in most jurisdictions in the world. We know that it has class 1 data in a number of different diseases, and most importantly, we know that it's extraordinarily safe, for at least as far as neurologic treatments go. But the question is, what part of the brain should we be stimulating? Because while it's not entirely focal, it's mostly focal, and an emerging body of data demonstrates that where you target matters, and it's not necessarily whether you hit the frontal lobe or not, specifically what part of the frontal lobe are you hitting. So this raises a question of how do we make sure that we're in the right place for the disease? And so again, this high-level diagram demonstrates what we're basically doing, where we take a structural atlas, we utilize the CST tractography to adjust the parcellation boundaries and adjust voxels to make a patient-specific atlas. And then we use that atlas to subset the resting state data and the CSD data to make adjacency matrices that we can then look at, use different machine learning tools to analyze and determine what is abnormal. Next. And again, one of the challenges with looking at this kind of high dimensional data is that you get a matrix like this. And if you look on the y-axis here, there's lots of tiny print, this is because there's 370 entries here. So, and you just start advancing. Yep, keep advancing. So as you can see, it's interesting to note that there's red and blue squares, but it's a lot more valuable when you can utilize prior knowledge to subset the data, either to base it on networks, base it on specific anatomy of interest, or eventually to make a disease-specific subsettings of the data. Excellent. So, for example, if I was studying a patient with a language dysfunction and I saw lots of anti-correlation in the language system, which we normally don't see, it's important to note that that column is a place in the brain, and it's area 55B, which you can see over there on the right. And there's some data that we have that this is a motor cortex for the larynx, or at least is involved in phonation in some kind of fashion. It's a speech area that's distinct from the normal canonical speech areas. So if you advance to the next slide, so this means that core connectivity can be leveraged in a way that interacting with the data, you can select a target. So it can be turned into a file that's used to treat with TMS, or it could be any other thing. We as a software are relatively agnostic to how the stimulation is done. We can just point out what is not supposed to be there, or give ideas on where the abnormalities are. So next slide. And again, going through the anomaly detector, we can go through this quickly because we covered this earlier. Black areas are areas that are highly variable, white are areas that are normal, and red and blue are areas that are more or less correlated with each other than normally seen in a normal range. The next one. And one of the things that is helpful is to And what we tried to focus on is how do we make it so that when we learn something, instead of being in a paper and so you may or may not come in contact with, that you may or may not be able to apply, that we can create a library that eventually, as we learn or other people learn, this can be leveraged on your own data. So how do you organize this? So we've created a wizard that we're in the process of filling in as much as we can, and we will continue to work on this, that can allow you to ask the question that you're interested in the form of a disease-specific question. So for example, if I were to pick depression in this wizard, just advance, advance, there are numerous tools that can be brought to bear. For example, looking at disease-specific states, classifying subtypes. These have all been described in the literature by different people. And so, again, it's not making a model that detects whether someone has depression or not. Again, that can usually be gleaned by talking to somebody. What we're interested in is what parts of the connectome would be specific for showing something like suicidal ideation. So the vision would be that eventually you could leverage this by saying, I want to look at my data in terms of suicidal ideation, and that it would look at the model that we have here and give you the appropriate subset for that based on machine learning. Next one. And this kind of segues into, I think, the end of our course, which we're going to talk about what is the long-term vision. And again, we think we have a very good processing pipeline. There's a lot you can do with it. It brings a lot to bear, particularly in being extremely anatomically specific and leveraging a lot of the data that we've been able to put into one architecture to speed up the process of really being able to leverage prior knowledge. But again, we weren't here to create a new FSL. What we're here to create is a methodology for mining this data and quickly being able to find areas of interest, methods for looking at this, and to not only be able to share that in quicker time and accelerate discovery, but to also to pipeline this into the clinic so that once something became clear, we could utilize this to eventually move things from the right-sided button into the left-sided button. And in that vision, if we had quality machine learning models that were able to look at depression and to go through it like I showed, where you could eventually develop a model that was robust, had a good AUC, that you could eventually have a transdiagnostic therapeutic approach to studying connectivity and to leveraging it into clinical practice. And what our long-term goal is, as these models become more robust to and able to withstand scrutiny, that we have a methodology that is eventually able to show this to clinicians in a way they can understand and leverage it. Next. And so in order to really have a model like that, it's one thing to talk about this as a vision, but how do you actually go about doing this? And this is some of the tools that we've created that are basically coming into maturity. And one of the things that my boss, when I was a junior faculty, pointed out to me is that the vast majority of patients don't make it to the research institute. They're treated in the hospital. And particularly some of the more interesting and sick patients are treated in the hospital. So if you want to study disease, we need to utilize the immense data collection that's going on in our hospitals every day to the best of our ability, instead of working through the more constrained version of formal research studies. So how did we do that? Well, one way is we utilize things like natural language processing to go through a medical record, figure out what do we want to see in advance, and to convert this into a database that can be utilized to classify patients, to identify potentially covariates that need to be regressed out of the data, etc. Another one that came up, next slide, is the fact that while a 5-hour neuropsychology battery can provide really outstanding information, this is not scalable. There's just not enough neuropsychologists in the world to see every patient that we treat, and thus we're passing on a lot of very valuable neuropsychological and clinical data by not having tools that are scalable to non-experts and in non-traditional situations. So for example, if I wanted to get neuropsychological testing on a glioblastoma patient who's scheduled for surgery tomorrow, probably they're not going to go through a 5-hour neuropsych battery, and that's an opportunity to learn that we've lost. Next. And so again, once you start talking about industrial grade data architectures, it's important that we provide the ability for people to put together specific queries and to handle the data in the cloud. If you're talking about turning a hospital into a data factory, you have to have the ability to go and ask questions on the data. And that can't be somebody taking a bunch of USBs and transferring data or putting things in Dropbox or any of the things that we've been doing, or even just having a loose AWS bucket. We really need the ability for clinicians and people who are not extremely savvy to easily work with this and say, I want to know this question on that data set. And so these are tools that we're in the process of deploying for working with this kind of data set to say, in the dream world, I want to look at everybody who has treatment-resistant depression who's been on an SSRI, and finding that data in your data set very quickly, and then eventually launching it into machine learning architectures for studying this at scale. So I'll turn this over to Stephane, and he can talk about some of the efforts on building the architecture to do what I said, which sounds grandiose, but is coming into reality in a very short time frame from us.
Thanks, Mike. So what we have here is just an illustration of where we see some of what we're doing going. In essence, while we capture some of the largest data set you can find on brain imagery, including as well neuropsychological measurements, as well as other elements you'd find in additional notes, demographic sheets, or whatever else, we also want to offer the ability for researchers to leverage that data effectively. And if you remember in the introductory part of this code, I mentioned the issues of running laptop scale analytics that should be run at a cluster scale. So running really enterprise grade machine learning in the cloud. And that's something that we're aiming towards developing, is basically a self-development kit, which would allow you to, within the platform, start prototyping any type of analytics to then be deployed and use our entire infrastructure to run it on the proprietary database we have, so that you can basically supercharge your research with way more participants that you would have gotten if you had to acquire them yourself. And what this slide essentially illustrates is something we're aiming towards, which is this future ability to go from a prototype code to an enterprise grade large-scale machine learning project, all using Omniscient's SDK to be released in the next few months. Michael, so if you want to take the conclusion.
So I think one of the things that we want to try to get across of what we're providing that's different, why we're excited about it, and why many people we talk to are excited about it. And ultimately, that coding is necessary to do neuroimaging research. But the answer is neuroscience and coding are not completely overlapping circles in the Venn diagram. And so we really want to bring back the idea that while we're doing sophisticated software analysis, that there also are people who are, really what their entire life is dedicated to is software engineering and enterprise grade architecture. And what we want to say is that the neuroscientists should be utilizing their skill sets to do neuroscience and not coding. Also, it's really important to note of how much machine learning architectures have advanced, particularly in the last 5 years. And what our goal is to accelerate the pace of learning, not only by making it simpler to process your own scans, but to utilize more federated methods like Stephane talked about to analyze data that's been uploaded by other people without having to go through all of the things about material transfer agreements and other things that about data ownership that make it challenging to collaborate together. And ultimately, the goal is to accelerate the pipeline of transitioning insights that we gain at conferences like OHBM into clinical insights that help patients. And so the idea that we want to have a 15, 20-year timeline to sort out everything about how the brain works before any of this translates into reality is really, I think, an idea that we need to move past. We need to think about how do we translate what we have as fast as we can, as soon as we understand it, into a reasonable level to help doctors. Because if you reality, having been a clinician myself for many years, if we don't provide at least some information, we're actually forcing them to work with minimal or no information. And the decision will be made whether or not we have that data or not. So we really, and I hope we really emphasize that we want to do better. We want to speed up the progress because the diseases are happening whether or not our research is ready for them or not. One project that we started working on that leverages this architecture is a project called the Glioma Connectome Project. It has multiple sites, numerous very prestigious sites that are interested in participating and agreed to participate. And this involves utilizing machine learning extensions of clinical data acquisitions to collect clinical connectomic and neuropsychological data. It spans multiple countries. In order to do a project like this and to get it done efficiently, you need enterprise-grade architecture. Otherwise, the cost of just passing scans around can be quite substantial. And so I hope that we've engaged everyone's interest in helping us work on this project. We as a company have thought lots of different ways of how to work with the research arm of the software, and ultimately we elected to have a free lightweight version of software available. This is what we showed you at the course. There are more advanced industrial grade solutions that we can talk about for larger groups, but we really wanted people to help work with us on how do we make the software better, more useful. We have development teams that can make changes in relatively fast order compared to what you're probably used to. And you can see the link to how do we engage and work together to make you facile with the software and to really start to get this up and running. So thanks for your time and thanks for listening. And of course, I'm always open if you have additional questions or comments. And I'm also happy to have longer, more detailed discussions with anyone who wants to talk more about what we've created and what we're working on. Thanks, everyone.
Thanks, everybody.